Thank you very much, Professor Kim, for the overly kind introduction. And first of all, I would really like to congratulate Sherry, David, Leslie, and the rest of the IOC for putting together such a wonderful meeting. Thanks for bringing the ISEF back to the Asia Pacific region. This is actually the first ISEF annual meeting after Kyoto back in 2019. And I still remember actually the first time when I still served on the board as a member at large that Melbourne was first proposed as a candidate city for the ISEF. I was still thrilled to see this actually come to true. This is wonderful, really exceptional. So my name is Han Hubert Yin. I work at this university called Tsinghua, based in Beijing, China. And we try to set up the link between basic research and applications. So I would like to have a disclosure statement also. So I'm also a co-founder and a shareholder of Carrier Biomed, which is also a round sponsor of this ISEF 2024. So in the, the frontier spirit of Australia, I would like to show you some of this newest development of our lab. So if some of this, a substantial portion of the talk today was first disclosed, disclosed for the first time, some part is not really polished, I apologized beforehand. So the focus of our lab is innate immunity, right? Especially after the 3 years COVID, we do know vaccine and all of other immunity response. So I still remember at the beginning of last year, I hosted Julius Hoffman from France at Tsinghua. So even after working in this field for close to 20 years, I still learn new things. Dr. Hoffman won Nobel Prize back in 2011 for the discovery of toll-like receptors. And you talk about toll-like receptor, of course, is a major part for our innate immunity. We're born with this self-protection. And in the immunological response, innate immunity could count for over 70 or 80%, so the majority. For you guys who speak German, you know, Toll in German means amazing, right? So these receptors, indeed amazing receptors. They're just like those guards on the watchtowers of the Great Wall, like in northern China. They would actually detect the pathogen or danger-associated molecular patterns and turn on self-protection. For example, the very first ligand for toll-like receptor is LPS. We have already heard a lot of stories associated with LPS in the last 2 days. So LPS is a component of the bacterial cell wall. And for people like you and me, we don't have cell wall on ourselves. So when we detect LPS, we know there's some alien invasion, if you will, and we turn on our self-protection. So apparently, if you have too little of this protection, you can't protect yourself. If you have excessive protection, you'd have autoimmune disorders. So to precisely control the innate immune response is the key. And since I started as an independent PI at the University of Colorado Boulder back in 2007, I've been working on just that, to develop small molecule probe for toll-like receptors. So our group reported the first small molecule receptors for toll-like receptor 1, 2, 5, 3, and 8. We also work on some of the agonists also, because those toll-like receptors, they are double-edged sword. You want to control these. And in the last, I would say, 4 or 5 years, we focused on this endosomal toll-like receptors, such as the 7 and 8, because there was no small molecule inhibitors for those crucial innate immune receptors before. So, for example, toll-like receptor 8 detect single-strand RNA, actually various associated single-strand RNA, just like SARS-CoV-2 that caused COVID. So we do know that excessive toll-like receptor 8 activation was associated with autoimmune disease such as rheumatoid arthritis. So we reported the very first toll-like receptors for molecular inhibitors, and in collaboration with the hospital based in Beijing, called Peking Union Medical College Hospital, we showed that could be a novel way to, as a candidate drug, to actually attack this autoimmune disease. Because even like we have biologics, there is still not very efficient and safe small molecule drugs for those autoimmune disease. We also tried some rare disease such as adult onset Still's disease. Even now, so I still got emails sometimes saying, Dr. Yin, I saw your paper in this Nature Chemical Biology. My sister got adult onset Still's disease. Can we get on your clinical trials? So there's definitely still much need for in that area. So as a basic scientist working in the lab, I sometimes underestimate the efforts to push things from bench to bedside. The paper was published back in 2018. What did we do in the last 5-6 years? We actually we worked with different teams for the safety evaluation, for the production, for all the pre-IND. Finally, last year, 2023, this first-in-class candidate drug was approved by US FDA as well as the Chinese CDE for clinical trials. So we just actually finished the first batch, single ascending doses, and shows pretty good safety. And right now, we're actually starting the multi-ascending doses for the evaluation of these new drugs. So for this toll-like receptor, what we talk about, we humans have 10 different toll-like receptors, just like TLR4. But EV apparently also play an important role in this cell-cell communication in the innate immunity response. Besides the surface receptors, such like TLR4, LPS is known also could be wrapped up by outer membrane vesicles and get into the cells, turn on program cell death such as apoptosis. And a co-PI in my lab, Dr. Yin Zhang, also asked the question, did EV, like many other things, we know extracellular vesicles were really perfect, really good at doing, also mediate the innate immune cell-cell communication. So this was published work, I can sum up really quickly. Yin showed that TR9, which was activated by ODN-DNA, which is a specifically methylated DNA molecule from the bacteria, could actually activate the cells, and in the EV secreted by these activated cells, they would actually carry out those cargoes, and upon uptake by the recipient cells, they would turn on the immunological consequence, and also by remodeling the cytoskeleton, actually have a feedback to regulate this important process. So this actually taught us maybe for this crucial, important innate immune response, toll-like EVs would play a crucial role in this cell-cell communications as well. And more recently, we also asked for bacterial EVs function in this cancer microenvironment modulation. We have already heard very exciting stories from many of our colleagues, including Yang Song, for example, on the bacterial EVs shows. Apparently, those bacteria live in the gut, have many different impacts in our health. And Dr. Jinyu Li in my lab worked with the Beijing Cancer Hospital, showed the bacterial vesicles facilitate colon cancer metastasis, especially the clinical relevant colon cancer liver metastasis. She also showed that the bacterial EV could actually have some efficacy to actually like suppress the immunological response, and also actually like may also actually suggest some like novel therapeutic strategy also. So here I want to show this movie because like I guess a lot of you guys already see a different version of this. I stole this from CCTV, which is a network in China, and this actually was actually a short movie prepared by NHK that Annabelle, Takahiro Chiesa-san actually helped to prepare that. So apparently, like actually this EV already penetrates to almost every single aspect of our biomedical research. In China, we found CSF back in 2017, and last year, I know we know that guy, right? So last year already, actually in the medicine division, actually over 40% of the proposals have some EV component. This actually is really quite impressive. Thinking about this, still like relatively young field because EV can do so many things. So a little bit like history. Again, as I mentioned, I started my lab back in 2007 in Boulder, and we started actually a Howard Hughes CIA project. That stands for Collaborative Innovation Award, but actually the abbreviation sounds interesting, CIA. But basically, we just try to develop a curvature sensor to actually sense the tension of the membrane surface and come up with a peptide or other probe for CSF. So a postdoc in my lab, Jonelle Salute, along with a graduate student Leslie Morton, started our efforts in EV. This was back in 2009. And Jonelle, after that, my lab and Jonelle's lab, after finishing postdoc in my lab, he became assistant professor at the Washington State University and then moved back to his home country, Philippines. So this is actually like what Jonelle's work in Colorado. Okay, this is a painting by another student in my lab, and later on at the cover of a journal, this tennis ball was what fluorescently like labeled peptides that specifically target EV. And actually, like it's interesting. So I went to visit Jonelle in March this year. Right now, he's the vice president of University Augustine at Iloilo, Philippines. This actually is a remote island, actually like 2 hours flight from Manila. But I felt pretty touched because I saw even his office actually it's almost like a reproduction of our office back in Colorado. And he set up a research center called C2B2 at this remote island in Philippines with a major focus on EV research. Right? You guys know that Philippines has a lot of like really interesting natural product from honey, from pineapple and things. So I just feel like this is where Iloilo is, at this region of Philippines. I was thinking really like our efforts like 15 years back in the States started to bear fruits, even touching like a remote corner of Philippines. So. And then, like as Dr. Kim mentioned, I'm in Tsinghua. So I moved from Colorado to Tsinghua full-time back in 2018. And again, EV remained a focus of my lab. So this is some more recent work. A student, Dr. Xue Jin, actually a student from our arch library, Peking University, reported LPS could actually stimulate, right? LPS is a TL4 ligand. To stimulate cells, and we saw each of its blinking is a secretion events of EV secretion. So she was asking the question, maybe for innate immunity, right? We know this is important, but too much immune response is no good either. So this could be a way to control excessive immune response. So when she looked into this, he found the interesting molecule PI4P, which is a phosphorylated version of PI, lipid, play an important role here. We know PI well. PI is the second messenger that regulate many important biological processes. So we're asking really what maybe the lipids also play a role here in this LPS-induced EV secretion. Talking about these, we sometimes, like yesterday, we heard the leader of the field talk about all of the omics technologies. There's so many different omics, genomics, proteomics, but really talking about lipids, I feel that we're still lagging behind. This is more like we're tell a war story. It's easy to focus on those general lines, but really like the people who went to the battlefield and fight, those soldiers. So lipids are indeed those soldiers in our war against different things. So we humans have more than 1,000 different lipids, and their abundance, their distribution, for example, PS, this negatively charged lipid molecules in normal cells, 99% of the PS pointing inward. Only when cells undergo apoptosis, for example, the PS cells flip out, and our immune system recognize them and clear them out. This is so-called eat-me signal, as many of us know. But coming to the lipids in EV, it's still hard to study. One of the reasons, like proteins, you can use antibody and other probing technique. For lipids, oftentimes you really have to look into their intrinsic characteristic, which is their molecular weight. So we work with chemists at Tsinghua, the Xia Yu's lab, trying to develop new chemical biology tools to study lipid. For example, we know where the double bond locates on the tail of the lipid matters because they actually really affect the shape of the lipid, and also the cis and trans isomers, they would have exactly the same molecular weight, but their function would be very different. So we actually use chemical biology method to form the four-member ring by different cleavage pattern. We can tell where the double bond is and what the cis-trans isomer is. We also actually use fingerprinting technique to study different phosphorylated location, really different regional isomer, if you will, that's a chemistry term, and study their different lipids function. So for these, PI could get phosphorylated ones, not only ones, but only with the dual phosphorylation also. And each of these phosphorylation or dephosphorylation step would be controlled by different enzymes also. This is a very dynamic, complex system. So actually this is indeed really hard to study. To make a long story short, we actually combined bioinformatics and biochemical method and showing that this PIP5K1C to phosphorylate PI4P to PI45P2 actually the P for LPS-induced EV secretion here. And this, because this was already published last year, I was make this a long, summarize the story. We think LPS could actually activate TL4 and turn on the interferon pathway. And by PIP5K1C, we recruit this thing into the endosome sorting and involve heat shock protein and the rat family, which actually was also known to play an important role in autophagy and other things. So that's maybe one of the ways to control excessive immunological response. They could actually facilitate EV secretion. So this actually may actually give us some clue how this excessive innate immune response was actually calculated, was actually regulated by EV. Remember, in the old days, we do actually think EV could be the trash bag. Actually, you throw out after dinner as one of the ways to regulate the immunological response. We talked about toll-like receptors so far, and toll-like receptor already won Nobel Prize back in 2011. So what's beyond these amazing receptors? In my personal opinion, I think Sigas-Lin pathway is really quite important. I still remember back in 2014, I invited my good friend, Professor James Chen from Southwestern Medical Center to Boulder to give a talk. The very first slide James actually presented was that immunity beyond toll-like receptors. So he was talking about this back then newly found innate immunological sensor in the cytosol. So unlike those toll-like receptor we found in the endosome, right? So far we talked about toll-like receptor 8, for example, they were found in the endosome. They're an ectodomain pointing to the lumen of the endosome. But the cGAS says double-stranded DNA, which actually usually is not actually like commonly found in the cytosol, and turn on the synthesis of cGAP, and then turn on the STING pathway. So far, we do know this could be a very essential pathway response, especially in the innate immune system. So we work with the James group on development of like novel therapeutic strategy to regulate cGAS STING as well. But so far, we haven't actually put anything into the clinical stage yet. So we think actually there's still like a lot of new things to study, even like there's already like, the cGAS was firstly discovered back in 2013, over 10 years ago. There's still a lot of new things. For example, we reported March 8 could be a non-canonical path to regulate this important group of innate immune receptors also. March 8, we reported last year, March 8 is the new E3 ligase regulating cGAS by putting the ubiquitination on the K63 sites, and it could actually like really like inhibit the double-strand DNA binding to cGAS. And we think this could be a way to a new way to get around the current failure in the drug development for the cGAS STING as potential autoimmune disorders regulation. So this paper was already published in Science Cycling, and actually was reported by the Science Journal also. So it just like shows how much we still need to learn in this important pathway, and this actually could actually have a potential in technology translation also. STING is the same. STING is downstream to cGAS. Last year, to that LPS paper, we published back-to-back in the Journal of Extracellular Vesicle about STING's excessive immunological response regulation. This was done by student Jiaqi Liang in my lab. So Jiaqi actually showed upon STING activation, both in the cell and in the EV, STING and phosphorylated STING level got significantly elevated. So he found this actually interesting because the STING actually like co-localized also with the multivascular body marker VPS16. So we were asking like actually like what we do know like actually STING undergo a lot like many different layers of regulation, right? STING actually upon femorization and further tetramerization could translocate between Golgi and ER. And we actually like found a protein called STAM, which is a part of ASPORT0. Actually, this again is interesting. Regulates the secretion of STING from like ER. I can get into the in the vesicle, and then eventually got secreted to the external of the cell. So we found actually like upon the STING, the oligomer, actually again, let's talk about the oligomerization, regulates the translocation of STING. STEM is induced, the expression level was elevated, and they basically push the STING out. So maybe again, we talk about this is important, but it's also very general. So cells got to have like multi-layers of control of these. So just to avoid excessive innate immune response, maybe this is the way for STEM, actually like it's a new mechanism, machinery to control excessive STING activation. However, as we just talked about, STEM is a part of the ASPORT0 pathway, and I'm sure many of you guys are very familiar with this secretion machinery. We found STING actually this function for STEM's regulation of STING is independent to the conventional path, conventional function of the ASPORT0. So when we first saw this, to be honest, we feel like actually like, we're pretty hesitated. Is this true or this is an artifact? And then luckily, after our paper was published in March last year, we saw a couple of new papers also reporting this new function of STEM. So I guess like the lesson we learned from this is that it seems like a a lot of these things could have multiple functions. Some of them actually we're even very familiar with could have additional insights that we need to explore. So to sum this up, we think EV also, besides cGAS, besides toll-like receptor, the cGAS STING pathway also is a very central and essential regulation mechanism to avoid, especially to avoid excessive immunological response. And this paper actually was actually one of the most viewed paper by Wiley in the first half of last year, just showing like the strong interest of EV like in general. Okay, cGAS STING, DNA, that's actually our machinery for to sense double-strand DNA. I used to work at this University of Colorado, Boulder. For maybe like work in the RNA field. Sometimes we got the nickname Boulder is the center of the RNA universe. So a lot of people actually around me actually like work on RNAs, and also we heard a lot of RNA story in the past couple of days already. So we had a joke in Boulder. They talk about the God was seeing three scientists, and God was like, scientist A, so what do you work on? And he said, I work on protein. And God said, wow, protein is so important. You got to sit on my left-hand side. And then the second scientist like, so what do you work on? Scientist said, I work on DNA. And God said, oh, DNA is even more important. You got to sit on my right-hand side. And then the third one, so what do you work on? And the scientist said, I work on RNA. And God said, okay, you take my seat. RNA is so important, apparently. And I still feel that we still actually need to learn a lot more things about EV-associated RNAs. So back in 2020, Dr. Catherine Xiao in the audience actually worked with Professor Ken Whitworth's lab, put up a perspective in JAM also, talking actually looking into speculating rather back then, I would say, different types of RNA shows in the EV-mediated cell-cell communications. And they could have actually many impact in different disease areas, such as aging, for example. Catherine showed that microRNA, mere 30B5P, actually was play important roles in regulating the cell senescence by actually regulating the third family proteins function. And recently we moved also to the double-stranded RNA. So this actually is I think is a very exciting field. We do know, for example, the Ming lab at the University of Pennsylvania showed EV-RNA affect innate immunity, and also from our lovely co-chair, Professor Ming Le's lab, Ming actually demonstrated very elegantly that RBC-EV carries double-stranded RNA. And we also talked about yesterday, this very tiny microRNA could actually render a lot of potential in therapeutic development also. So Gaoge Sun actually also sitting in the audience. By the way, Gaoge also presented a poster number 155. I would encourage you to see this excellent young guy and talk about his work also. He was asking, besides red blood cells, how about platelets? For EV, I guess you can't really get wrong with platelets, right? In healthy people, in our body fluid, in the blood, a major source of the EV from the platelets. Even in the field of hematology, people still actually like to prefer using the word microparticles, indicating that platelets-derived particles that actually regulate blood clotting process and things like that. So in cancer patients, if you're aware, actually one of the leading lethal cause is blood clotting. So they just actually tend to clot more. And we're actually asking the question really what EV shows in the tumor cells communication with platelets. Platelets is an interesting cell because they don't have nuclei. They can't actually produce proteins themselves. So when Gauguin looked into different patients' sample, especially by different RA, he actually found it interesting that the RPMRA, which is the component for the ribosome was made of different protein and RNA molecules, and for RPMRA, actually was, we still know so little about them. It seems in different cancer patients, the RPMRA underwent substantial decay. And he was looking at different, actually like the RPMRA sequence in the platelets, and they all show actually significant decrease. So Gauguin was trying to collect some of the cancer cell-derived EVs, and actually to see the impact on the microkaryocytes, because again, the platelets do not have the machinery to produce the proteins, and they were produced by the other cells. And seems as a consequence of that, this EV's treatment would cause platelet activation. We thought platelets, actually the resting state is actually rather this regular shape, and upon activation, it actually underwent a pretty dramatic shape change, and it triggers the blood clotting process. So we thought this actually is really quite interesting. Again, we already talked about the cGAS-STING pathway, right, as a double-stranded DNA sensing machinery. Gauguin was actually asking the question, what about double-stranded RNA? It seems actually in the presence of ATP and also the OAS, that is in parallel to the cGAS-STING pathway for DNA enzyme that also synthesize these RNA cells that will actually triggering on the RNA decay process. So we found maybe it seems like in the presence of the double-stranded RNA, the tumor cells use that to actually figure again to tell there's something changed, and then I want to actually as a consequence for the immunological self-protection machinery would actually trigger the decay of mRNA. So we found this it is really quite interesting, and the tumor EVs seems, we do know, for people who work on RNA, you would know this is much more unstable than DNA. During the COVID period, when we do the environment test, DNA, RNA usually just was gone after several weeks. DNA could last sometimes for a year or so, and it's really caused a lot of pain, especially when we did a lot of these nucleic acid tests during the COVID. So what we found, EV actually protect this extracellular RNA, and of course the free RNA got degraded, and those EVs were indeed actually taken up by the cells. And we think this actually is interesting. Also, we coined a term called the tag up for this process, not only because this actually shows a interesting RNA, double-stranded RNA shipping mechanism, but also actually may actually a rather non-conventional biomarker strategy. Usually we saw the different level of elevation of the markers, but for this case, maybe the RPMRA's decay could be indication for the disease such as tumor. Then how about mRNA transportation? Another student coming to this ISF 2024, Haixiang Zhang, also was asking the question, really what the machinery help to actually pack, sort, and secrete those messenger RNA, right? RNA has all different types. So there actually were interesting previous studies previously, but the specific machinery how to tackle mRNA for the EV secretion was still actually in elucidation. So Haixiang actually after screening, he identified a RNA binding protein, or RBP, called NPM1, seems relevant. So in this figure, you can see that NPM1 not only co-localized with RNA molecules and also particle markers. It seems actually they even like wrap this up. If you knock down NPM1, the mRNA, the EV-dependent mRNA transportation basically got blocked. So we found this actually is interesting. This actually could actually render some like the first insight of this sequence-specific RNA transportation shipping machinery. Again, by combination of bioinformatics and biochemical method, Kai-Xiang actually identified those candidate sequences. Actually, they're short. This actually, again, I think it's exciting also for potential technology translation, because when we attach those sequences to different mRNA, they could actually ship mRNA, push mRNA into the EVs. Could be a interesting strategy to ship those important nucleic acid molecules. So why did NPM1 like do this? So we think it seems like this mRNA sequence was often found in different oncogenes, basically in the pathogenesis of cancers. So when we look into those sequences, seems they actually like, especially in the different like carcinoma, for example, they were found. Maybe that's a way for cancer cells, like urgent needs, to talk with the environment and shipping the messenger RNA out. So we showed this mRNA indeed actually get into the EV, not actually like laying on the surface. They are not, I guess another, what's the term, like yesterday people talk about surface like thing, but actually they were really inserted into the EV. And also this oncogeneous mRNA could be taken up by the cells. So we also showed this actually is relevant in clinical samples. Kai-Xiang collected samples from like patients with different cancers at different stages. It showed actually like compared to the normal EVs, the cancer patients really use this actually EVs, NPM1 dependent pathway to transport messenger RNA. So we speculate this could be a way for cancer really to stimulate the environment, actually affect for long distance communication and helping the cancer spreading basically also. So upon that, coming back to technology, we talk about the from bench to bedside. Back in 2009, we first worked on the curvature sensor. We tried to actually like build a peptide that senses particular curvature and lipid composition. Again, for curvature, we also already heard some discussions at this meeting already. So you can define, for example, at this point, the curvature is that you fit a sphere, like actually a small ball, snooker ball, for example. The smaller the ball is, the more curvature you have on the surface. You can just define this as the reciprocal of the radii of the ball, basically. So, for example, like a point like this, a combination of C1 and C2, different curvature, would give you a different shape. And we do know, apparently, in many biological processes, including EV uptake and secretion, membrane curvature play crucial roles. So, this is an AI time. I feel like almost every single meeting, no matter what area you work on, people talk about computer science. So back then, a MD PhD student in my lab, Noah Castillo-Ruiz, right now he's practicing medicine in Palo Alto at Stanford, oncology. So back then when he was a student in my lab, he used MD simulation, the molecular dynamics simulation, and while he was pursuing an MD degree, to show that with a highly curved membrane, just like EV, right? They're small, and they also have, because the small size, the lipids do not actually pack as well. They would actually give you the defects. So we're thinking, based on this characteristic, we can rationally design novel probe for these EVs, and really trying to grab the most disease-relevant EVs with small size, with a particular lipid component on the surface. So after I moved to Tsinghua, we also collaborated, have international collaboration with Janelle. Remember this guy who actually locates at Ilo Island in Philippines? We tried to actually use the first generation of the peptide probe for curvature sensor to actually detect this highly curved and lipid-specific curvature of different EVs. So this actually was, we have a international collaboration project, and this was when Janelle came to Tsinghua last year. And also actually I saw already, I have some the third generation, I mean the Philippine local student, they also got really strong interest in EV also. That's really encouraging. I feel like this, the academic line, the EV interest keep passing on. So for this, we just like to, because it was published, for the sake of time, I would like to quickly mention, we have this fluorescently labeled peptide that specifically binds to a highly curved curvature of EV. For example, we're thinking this compared to the conventional ultracentrifugation method, it's much easy to use. So, for example, this movie doesn't show, but this is a pathological rat model of blood. We can show in this complex environment, we add fluorescently labeled peptide to show on this figure only those small EV actually got labeled. So this could be a way for quick and economic alternative to the EV detection method. And we work with entrepreneurs also. Dr. Fred Lee, actually, sitting in the audience, we co-founded Carrier Biomed, based in Suzhou, which is an industrial city about 100 kilometers away from Shanghai. So we try to actually mobilize those curvature sensing peptide on the magnetic beads, and then it's actually pretty easy to use. Last year, we also came up with a machine that is fully automated. You can actually put the patient's sample in, and EV got out quickly. So even me, after 20 minutes training, I can actually use that myself. It's pretty easy to use, I can tell. And then we work with clinicians for clinical trials. IIT, as I mentioned, Carrier Biomed is the sponsor of this meeting. So also, basically, also in collaboration with Leslie, Professor Annie Hill, and other clinician group in China, we work on different neurodegenerative disease and various cancer. So my time is running out. I'm not going to show you too much clinical data. We showed in different cancers, such as lung cancer, we can differentiate different stages, and also between patients from inflammation group and normal group. And for some other cancers, we actually can follow those cancer patients, not only the static snapshot rather. So indeed, we hope this thing could actually provide useful information to the clinicians in their clinical practice. Finally, we disclosed the results for Alzheimer and Parkinson's disease molecular diagnosis. Actually, we tested blood EV in a group of more than 700 patients. We showed about 86% of accuracy, which is comparable to the gold standard currently in the market. So finally, I mentioned the people who actually did the work. Finally, thank you very much for your attention. Thank you.
Yes, this is a great talk. How successfully he bridged the basic science to biomedical application. I'm sure you have many questions, but so I will take only one question. Okay.
Thanks for a really inspirational talk. I've got a very naive question about the innate immune system. It's an inflammasome question. So there's this ubiquitous machinery in the innate immune system, and there is a connection between the toll-like receptors and the inflammasome. Is it known whether there is EV machinery involved?
Oh, that's a great question. Thank you for giving me the next proposal topic.
I give it to you.
On the top of my head, nothing came out right this moment. But this sounds like part of
It's
something
got,
it's not.
it has to be involved, yeah.
Actually, pyroptosis. Actually, I want to say pyroptosis, for example. I mean, again, this actually was done by my good friend Professor Feng Shao at NIPS in Beijing. They talk about actually this is even more severe than apoptosis. When when cells undergo severe infection, they program they burn themselves out rather, even more severe. I think there's some bubble particles actually in that process. Maybe suggesting this could be a very interesting direction we need to look into. Thank you. That's actually a great idea. Thank you.
Please give him a big hands again for his wonderful presentation. So next speaker will be chaired by Professor Le Min.
May I introduce Dr. Ryo Kojima from the University of Tokyo, Japan. He will share with us about barcoding of small extracellular vesicles with CRISPR-Cas9 to enable high-throughput subpopulation-specific analysis of their release regulators.
Good morning, everyone. So my name is Yosuke Kojima, and I'm Associate Professor at the Graduate School of Medicine at the University of Tokyo. So it's a great honor to be, for me to be able to speak in this session. So I really thank all for selecting me for this important topic. So today I'm going to talk about using the gRNA barcoded EVs for understanding their biology. So probably due to the limited time, I can skip most of the introduction about the EVs. So I hope everyone agrees that understanding of EV release regulators is important from various viewpoints, including advancing biology and also clinical purposes. So conventionally, if people want to assay what genes are important for SCB releases, typically I can just people just see the cells in the separate wells and then perturb intracellular pathway with some small molecule or siRNA, one by one, and then assay the amount of SCB released in each well by some manner. But I think the throughput is too low to comprehensively understand complex disease processes of SCBs. So then our idea is to use barcoded SCB. So if we can encapsulate barcode nucleic acids encoding the information of gene knockout into the CVs, it would be possible to conduct full assay of the amount of EV released by checking barcode composition by NGS. So in one flask, so we prepared a pool of cells in which each in which different genes are knocked out respectively, and let the cells to release SCVs. And for example, in this kind of case, we can guess, okay, maybe knockout of gene C might decrease the amount of SCV released, or knockout of gene D might increase the amount of SCV released in a pooled manner. So that's the idea. So then the question is technically how we can create such barcoded EVs. For this, we have previously developed so-called exotic device for actively loading RNA into SCVs. So concretely, we fuse an RNA binding protein called L7A to EV marker CD63, and at the same time, we incorporated CD box which binds the L7A, and then the active interaction of CD box and L7A enables to actively incorporate RNA into EVs. And several other researchers have also reported some other combination of RNA binding protein and motifs can be used for this purpose. So in this study, we combined this system with CRISPR-Cas9 system, as shown here. So for the CRISPR-Cas9 system, Cas9 and guide RNA are co-expressed in the cells, and then target gene is knocked out. And we, at the same time, co-expressed the DCAS9 fused with CD63. So then DCAS9 works as the RNA binding protein against the gRNA, and then this gRNA is encapsulated in EVs. So this gRNA encodes the information of the gene knockout in the originating cells, so this gRNA can be used as a barcode I mentioned. First, we tested whether this CD63-DCAS9 enables efficient journey loading into EVs. So without CD63-DCAS9, almost no transcripts of journey was detected in EVs, but by expressing this component, we could clearly see the copies of journey by qPCR. And for the mastery parallel analysis of journey by NGS, we need to have some method to amplify journey in a parallel manner. But one thing to consider is spacer sequence against target of the genome is located at the 5' of the journey in the case of Cas9 system. So normal PCR cannot be applied. So for this purpose, we apply template switching method to incorporate some common sequence at the 5' end during the reverse transcription. Combining this nested PCR, we succeeded in amplifying journey-derived amplicon as shown here. And next, so we infected journey library in the format of lentivirus to CD63-DCAS9 expressing cells and extracted journey from both cells and SEVs as shown here. And this is a graph showing that in x-axis, so we tested more than 20,000 guide RNAs and ranking is here, and y-axis is normalized read count of each guide RNA. And as shown here, we succeeded in detecting about 90% of spacer sequences by NGS with low skew ratio in both cells and SEVs. So if we compare the journey abundance in both fractions, a clear linearity was observed as shown here, so which indicates that guide RNA can be actively incorporated into EVs without being affected by the spacer sequence. And the reproducibility of the barcode decoding is very good. So having these prerequisites for screening set, so we conducted screening for extracting the SEV regulators, which we call, the pipeline is called now cyber screening. So we prepared Cas9 negative cells and Cas9 positive cells, and both of which express CD63-DCAS9, and led the cells to release EVs. And basically, we can just compare the composition of guide RNA inside EVs. And for example, in case of, green guide RNA, maybe knockout of the green gene might increase the amount of SEVs. So we can guess this kind of thing in a parallel manner. But one thing to consider is knockout of some specific gene can affect cell viability or transcription of barcode. So we need to cancel out. So and then in this case also barcode inside EVs is not detected. So we need to have some algorithm to cancel out this kind of effect to extract it through SEVD dysregulators. And for this there this kind of algorithm. So concretely, we first from the normalized read count of each guide RNA from both cells and EVs, we calculated fold increase of each guide RNA abundance in both cells and SEVs, which is shown as the FC cells and FC SEVs here, between Cas9 plus and Cas9 minus condition. And when we plotted FC cells and FC SEVs, as shown here, the linearity was observed, so which indicates that the change of the amount of each guide RNA is highly dependent on the change of the guide RNA amount inside the cells. So we conducted DNA regression here of FC cells and SCFVs, and defined the SEV release effect as the error term against this line. And because we are using multiple guide RNA against one gene, so we took the median value of each release effect of each guide RNA as a representative score of each gene. And with this setup, knockout of the genes having positive release effect values are thought to increase the amount of SEV released, and also knockout of the genes having negative values are thought to increase decrease the amount of SEV released. So we use this pipeline for the assay. So we conducted screening with commercially available guide RNA library, and we detected upper hits and lower hits as shown here. And first, we want to emphasize that CD63 was detected as the strong lower hits, which is quite reasonable because knockout of CD63 also knocked out CD63-DCAS9, and then guide RNA inside EVs depleted. So positive control is sort of working. And as shown here, so many genes were found as the upper hits and lower hits. And for example, FASN and PI4K, because we found a small molecule inhibitor against these genes, we conducted validation with these inhibitors, and we treated the cells with the inhibitors against these genes and monitored the SCB released from the cells with the reporter assay. And indeed, we could confirm inhibition of PI4K drastically increases the amount of SCB released, and while inhibition of FASN suppresses the SCB release. So we succeeded in finding true SCB release regulators. And we also conducted orthogonal validation against other genes with siRNA, and generally the amount of SCB changed as predicted by our screening. So these data demonstrate that the genes regulating SCB's release are highly enriched in the cyber screening hits. So due to the limited time, I cannot explain the biological finding we found with this assay scheme. So I just want to know what we did in this screening pipeline. But we want to know that this screening can estimate the release effect of all genes tested. So by using the score of each gene, various informatic analysis can be conducted, such as string analysis or gene set management analysis. For example, we can visualize the network of the hits genes as shown here. And also we can conduct gene ontology analysis or gene set enrichment analysis. So this screening pipeline enables to detect functional gene sets that affect the SCB release processes. And I also want to mention that the small EVs are heterogeneous, and everyone, I think, agrees that how the release of different SCB subpopulation is controlled remains poorly understood. So we wanted to address this point by using our screening pipeline. So for this purpose, we conducted another screening with CD9 as another protein to fuse with dCAS9, and compared the result with our CD6SD based screening as shown here. So in case of CD9 screening, again, then CD9 was detected as the strong lower hit, which indicates that our ball coding strategy is working. So it can be flexibly adjusted by choosing the appropriate SCB marker. And PI4K and FASN were found to be common regulators for both populations, but genes around here and here should be act differently on different subpopulations of SCBs. So so I just want to highlight one pathway which we found that affecting different subpopulations of EVs. We focused on the effect of cell cycle. So this is a result of GSEA analysis, and each dot represents the gene set. And the inhibition of gene sets shown here is supposed to decrease the SCB released amount released from this CD6SD subpopulation, and in this case CD9 subpopulation. And stopping cell cycle was suggested to decrease the SCB release amount only in the case of CD9 SCBs. So we synchronized the cell cycle by double TMD block and released the cells after synchronization of the cell cycle, and found that only in the case of CD9, correlation was observed with the cell cycle progression and SCB released amount, and the release of CD9 positive SCB seems to reach the maximum when most of cells had completed cell division. So in summary, we have developed cyber screening that enables to detect SCB release regulators in a massively parallel manner by using SCBs backlinked with gRNAs, and effects of more than 10,000 genes were assayed, and we have confirmed that true SCB release regulators are concentrated in the screening hits. And by changing the dCAS9 protein, it's possible to screen SCB release regulators in a subpopulation-specific manner. So we couldn't introduce the detailed result about the biology we found with this screening, but we already posted the paper showing this pipeline in the archive, and now the paper is in revision. So if you are interested, please just access to the preprint, and I would be really happy to get some feedback. And I think it would be interesting to compare the assay results by using different cell types, and also we are interested in using this gRNA barcoded EVs for other purposes, like tracing the EVs fate by tracing gRNA in the future. So that's all. Thank you. Sorry, I just need to show an acknowledgement. So this work was done by a very talented student, Koki Kunitake, and was supported by many other collaborators and also the funding source. So thank you for your attention, and I would be happy to take any questions you have.
We have time for one question, one quick question.
Really outstanding work. I have a question about you observe differences changing the receptors, not the or you're producing the guides, but I'm thinking about two more microenvironment crosswalk. I'm thinking of differences taking up. It is one of the hands when you could have macrophages who are able to degrade most efficiently, and maybe other cells are not that efficient. So have you tested this with different receptor cells in order to see if the screening is changed?
So you mean so treating the barcode cells to the macrophages and how they're eating?
Yeah, either macrophages or epithelial cells, so two different receptors in order to see the machinery that is receiving in the cell could affect the outcome.
Okay, yeah.
I guess it could, but if you have tested.
Yeah, so we are now testing those kind of trials to trace the gRNA also for the uptake of the cells, but it's also technically not easy to detect the barcode from the tons of RNA inside the receiver cells. So we are now struggling to improve our system to achieve such kind of application as well. Yeah. Thank you.
Please join me to thanks for working here for two excellent talks this morning.